we are going to learn aminoglycosides. So let me give some small introduction about aminoglycosides. So aminoglycosides are named because the structure contains amino sugar, that is your glycone part containing a glucose ring, okay, amino sugar linked by a glycoside being glycosidically to a aminocyclitol. What is this aminocyclitol? It's a cyclohexane ring containing your amino groups and OH groups. So that's the glycone part of your aminoglycosides. So they contain an amino sugar, that is a glycone part, which is linked glycosidically, that is O-glycosidic linkage with your aminocyclitol, that is your glycone part. They are also called as aminocyclitol antibiotics. Why? Because it contains a cyclic ring, hexagonal ring, cyclohexane with OH and amino group. That's why they are called aminocyclitol. So aminoglycosides are also called as aminocyclitol antibiotics. Why? Because it contains your sugar as well as your aminocyclitol. Okay, glycone and aglycone both are present in this aminoglycosides. They are mainly active against gram-negative bacteria, which the gram-negative bacteria include model organisms like Escherichia coli, as well as your more pathogenic bacteria such as Pseudomonas aeruginosa, Neisseria gonorrhea, Chlamydia, Trachomatis, and Yersinia pestis. So these are the spectrum of activity. Let's go further. So the spectrum, as I said, it is most active against gram-negative bacteria. Aminoglycosides are broad spectrum activity. They are used in systemic infection caused by aerobic gram-negative Bacillus Klebsiella protein enterobacterus, tuberculosis brucellosis, tularemia, Yersinia infections, amoebic dysentery, Shigella. So in your stomach infection also they can be given. Pneumonia and urinary tract infection, it can be given. As you know, streptomycin is given as an alternative first line therapy. Okay, gram positive and gram negative anaerobic cocci include, except your staphylococci and your anaerobic bacteria are less susceptible, though it is broad, but they have got limited in case of gram positive bacteria. Now let's go for the classification. So aminoglycosides can be classified according to the species from that. If it is obtained from Streptomyces species, the suffix mycin comes, MYCI. If it is obtained from Monospora species, then the suffix MICN comes. See the difference between the species from where it is obtained. Example of Streptomyces is mycin species are Streptomycin, Paramycin, Neomycin, Tobramycin. Whereas in case of obtained from your Monospora species, suffix MICN, it is gentamicin, amikacin, and netilimycin. So this is the broad classification according to the source. Now the classification according to the route of administration. Topical, that is eye and skin, neomycin, prasetin, that is soframycin, gentamicin. Systemic, streptomycin, netilimycin, gentamicin, tobramycin, kanamycin. GIT, it is neomycin, paramycin, and this can be used in your GI infection. So this is the classification according to the route of administration of your drugs. Okay. Now let's go for the mechanism. So what's the mechanism? So this is your gram-negative bacteria cell wall. It contains your cell wall, that is a lipoprotein membrane, a peptidoglycan, and the plasma membrane. So there are porons which are present on the cell wall. So there are porons which are present. So this aminoglycosides pass through these porine channels passively. They pass through the porine channels with the pores which are present in your cell wall. Okay, this is through passive diffusion. Okay, further transport across the cytoplasmic membrane takes place through an active transport by proton pump, that is oxygen dependent. Through against the gradient, it will go through the cell walls. Okay, this is how it enters the nucleus. So once it is inside, it will bind to the 30S ribosomal subunit. So once it binds to the 30S ribosomal subunit, they interfere with the initiation process, that is your messenger transfer. So it inhibits the transfer of messages from your mRNA. This induces misreading of genetic codon on the mRNA. So there will be no propagation, chain propagation. Your codons will not be synthesized. There will be misreading of. This leads to the breakup of your polysomes into monosomes. So this will lead to your breaking of your polysomes, means long chain of amino sequence which will be present, nucleotide sequence, that will be broken into monosomes. So once this polysomes are broken into, so the protein synthesis is blocked. Okay, so this is the mechanism of action. Now let's go for the mechanism of resistance. That's the what will be the sources of resistance? Synthesis of plasmid mediated transferase enzymes. So these enzymes, if you see amino acids, when we'll be studying the structure, it has got lot of amino and OH group. So this OH group and amino groups are prone to your acetylation, acylation, different types of reaction, which is mediated by bacterial transferase enzymes, which inactivates your glycosides. Okay. Now, decrease transport into your cytosol. So that energy dependent active transport, that can be blocked by your microbial organisms. So they will inhibit your active transport across the cell membrane. Okay. Deletion or alteration in the receptor proteins of your 30S. So one thing is that there will be some modification, mutation in the sequence. So that will prevent the binding of your amino acid on the 30S ribosomal by mutation, and thus prevents its attachment. Once the attachment is not there, so aminoglycosides cannot recognize your site for attachment, and thus lead to the resistance. So as we explained, one of the mechanisms through deactivation through enzymes. So why this many resistance has attained to many of the species, like against streptomycin, kanamycin in clinic. So there are R factors are responsible for your production of aminoglycoside deactivating enzyme. So this R factor which synthesize enzymes which deactivate your amino acids. So which are the enzymes that deactivates the aminoglycosides? That are acetyl transferase, which can transfers acetyl group, phosphotransferase, which transfers your phospho group, okay, to it. So that is given by ACC, is your acetyl transferase, APH, that is phosphotransferase, and nucleotide transferase. Okay, this transfer nucleotides, that is your adenine, guanine, it transfers to your OH present in your aminoglycosides. These enzymes transfer to hydroxyl and amino groups that are present. So all these groups, acetyl, phosphorus, can be transferred to your OH and amino groups by this enzyme. So let us see the sites. So where does this acetyl transferase act? They act on the third amino group in the second ring, 2-6- amino groups in their ring 1. So this is the site of action. APH, phosphatase, on the third OH in the first ring and 2 OH in the third ring. And your nucleotide transferase attacks on the second and fourth OH of your third ring and the fourth OH of your first ring. So these are the sites of action of this aminoglycoside, of this enzyme. So these are all the sites, OH and NH2. These are very much prone to your acetylation, phosphorylation, nucleotide translation. Okay, so these are the sites. So these are the enzymes which act on this specific sites. So please remember this thing. Now let's go for the chemistry. So basically there are 3 parts in your aminoglycosides. One is your sugar, which is connected to the glycone part, that is your aminocyclitol, that is deoxystreptamine or streptidine moiety. That is your glycone, but again connected by O-glycosidic linkage to your amino sugar. So there are two amino sugars which is connected to a glycone. Only difference between your aminoglycoside structure and streptomycin is there is a streptidine, that is followed by a streptoamino sugar and N-methyl glucosamine amino sugar. So these two together it is called as streptobiosamine. So only the sequence changes in your streptomycin. Now let's go for the chemistry. So cyclohexane so if you see, as I said, it contains a aminocyclitol. That means it contains the cyclohexane group with several substitution or unsubstituted amino and hydroxyl group. So the cyclohexane contains several substituted as well as unsubstituted amino acids and hydroxyl, which brings high water solubility. Because of this presence of this amino and hydroxyl group, this increases the solubility of water solubility. So some of the examples of your aminocyclitols are streptidine. So if you see, streptidine means you see the at 1 and 3rd position there is an guanidine type of linkage. At 1 and 3 there is a guanidino type of linkage. It's a digonide. In case of streptamine, at 1 and 3 position there are amino group. They are known as streptamine. So remember these two. Most of your aminoglycoside contains these two groups. Okay, most of the aminoglycoside contain 2-deoxystreptamine means there is no OH at the 2nd position. 1 and other position contains your OH. Next is streptamine, that is your N-methylated. There is no NH at the 1st position and OH at the 6th position. Photamine is at the 1st position there is an amino group, and at the 3rd position there is a methoxy and N-methylated. So most important things are your deoxystreptamine, streptidine and streptamine. So these are the 3 most frequent amino acids, aminocyclitols which are present. Along with this thing, they have an aminohexose as an amino sugar, and some has pentose as extra. So some of the aminoglycosides may contain extra sugar moiety also, ribose sugar also. Okay, pentose sugar. Basically they contain aminohexose sugar. That means 6-membered sugars with amino group in it. So this is the basic chemistry of your aminoglycosides. So now let's go for your SAR. So there are 3 rings. Okay, sugar, first amino sugar, second is your cyclitol, third is your again sugar moiety, amino sugar. Okay, now let's go one by one ring. Okay, let's go for the ring 1. So let's go for the ring 1. So it is important for your broad spectrum activity. This is necessary for your broad spectrum activity. The 2nd and the 6th amino sugar. The 2nd, if you see the amino group at your 2nd and the 6th, don't get confused with the structures, just remember, okay, because the structures keeps on changing with different moiety. So the 2-6- amino function group are important for your activity. The 2- and the 6- amino groups are necessary for your activity. Okay, methylation of your 6th amino group, okay, does not lower the activity. Okay, methylation or of the 6 does not have lower the activity, so it does not have any effect on your activities. It also confers the enzymatic resistance of acylation on the 6th amino. So this methylation also prevents the acylation, so your resistance decreases. Okay, toward the removal of the 3rd position OH and the 4th position both in kanamycin structure does not lower the activity. So if you remove the OH at the 3rd and the 4th position, the activity is written. Why? There is no change in activity, but your resistance across to the deactivating enzymes increases. Okay, so they will not get. So 3,4-deoxy, that is kanamycin, is also potent. Gentamicin series lacks OH functional group at the third and still the compounds are active. As we said, third and fourth position, if the OH is removed, still the activity is maintained. In case of sisomycin, a double bond is present between the fourth and the fifth. See these structures, fourth and at the first ring. This still they are not, and because of this they are not deactivated by your phosphotransfer. So this all positions, if there is a double bond between 4 and 5, 3 and 4 hydroxyl groups are removed, methylation at the 6th position, still they are necessary for the activity. They do not remove the activity. Now let's go for the SAR at the second ring. Okay, it is the flexible change. So one amino group in amino acid can be acylated and antibacterial activity remains unchanged. So even if you acylate at the first amino group, but resistance against DIT increases. So if you acylate at the first position amino acid, your resistance against your DIT enzyme, example amikacin. So remember all these things, because when we read the structures one by one, you have to recollect your SAR. The one amino group in sisomycin is ethylated, saves the antibacterial activity and increases the enzymatic resistance, that is netillamycin. So in case of netillamycin, there is your amino group at the first position is ethylated. Your amino group at the first position of your aminocyclotol is ethylated. So this increases your resistance against the enzymatic resistance. Now the third ring is also an amino sugar. Okay, they are less sensitive to modification, so there might be many changes that can be done on the third ring. So 2-deoxygentamicins are less active than 2-hydroxy ones. Okay, the 2- amino groups in case of sildomycin are very active. So if you replace your OH by NH2, the activity increases. The amino group can be a there is a amino group at the third carbon. So that can be a primary or secondary. Most important thing, the OH at the fourth position can be axial or equatorial. The former one, the axial one, is resistant against the deactivation. So when the OH is in the axial position, there is strain. So that is resistant to your deactivating enzyme. Okay, nucleotide deactivating enzyme. So these are the SR. These are the important things in your aminoglycosides. SAR of your aminoglycosides. Now let's go see the overall. So these are the 3 rings. Okay, if you see 1, 2, 3 rings. Okay, these are the 3 rings which are present in aminoglycoside. So first ring, what did we say, everybody? So NH, it contains NH2 at the second and the 6th position. If you see the structure, ring 1 has got NH2 at the second and the 6th position. If you see over here, okay, NH2 at the second and the 6th, that is necessary. Now, in case of gentamicin, carbon 1 and 2 has different patterns of methylation at your 6th position. So in gentamicin, there it is R is methylated. Deamination also will exhibit good active against. So if you remove the amino group at the 2nd position, the activity is still removed. Lack of amino group at the 3rd and the hydroxyl group compared to is replaced. So if you remove the NH2 and OH, still the activity is retained. Hydroxyl group can replace the amino group. They may remove the activity. So amino group is necessary. If it is hydroxyl, it will reduce. Okay. Double bond, if it is present between 4th and 5th, the activity is maintained. In the case of the second ring, deamine will exhibit good activity against certain bacteria. If the amino group at the 3rd position is removed, still the activity is maintained. The hydroxyl group may be resistant. So always it is a deoxystreptomine and streptidine which is used, because removal of your oxygen will OH group will activate your increase your resistance against your deactivating enzyme. So at the second ring, the deoxystreptomine which is present in the is more effective. Now at the first position it can be ethylated or acetylated to prevent your deactivation by your enzyme. Third ring. Now let us go for the third ring. Okay, your OH should be always axial. Okay, it is axial. Okay, it should be axial, or it can be fourth OH can be axial. Second OH might be an hydroxyl or an amino group. Your amino group at the third position can be a primary or a secondary amine. So both can be, and the fifth there is no CH2. Okay, it can be an sugar. So these are the basic structural activity relations that has to be present in your aminoglycosides. Now let's go for one by one example. Now let's go. First example is kanamycin. The first example is kanamycin. So you draw an sugar moiety, that is an O with an cyclohexane ring. Okay, give the numbering. Next to your down to your there is 1, 2, 3, 4, 5, and 6th is your CH2R1 group. So CH2R1 group. Second, it is connected by O glycosidic linkage to the fourth carbon of your aminocyclitol. Remember, it is connected to your fourth carbon of your aminocycle, that is your aglycone part. Okay, the aglycone part over here is 2-deoxystreptomine, that is NH2 at the first and the third position, then OH. So there is a OH at, there is no OH at the second position. Now it is connected by O glycosidic linkage at the sixth position to your another sugar, amino sugar. So let us see what is your amino sugar that is present in your third sugar, amino sugar ring. So it has got an OH. So it is attached to the first position by O glycosidic linkage, which is connected to the sixth carbon of your deoxystreptomine. Now at the second position there is an OH. Third position it contains amino group NH2, plain NH2. Fourth contains your OH, which is mostly axial. Fifth position is connected to CH2OH. Okay, CH2OH. Now let us see what are the substitutions. So at the sixth position of the first ring, it might contain an, so what is R1? So R1 is NH2 and R2 is OH. So in case of this thing, your sixth position is an amino group and your second carbon contains an OH. So this is what is kanamycin. You have to remember basically kanamycin A. It is always present in the, so you have to remember the structure of kanamycin A. So kanamycin A contains what? In the first amino sugar, it contains OH at the second position, OH at the third, OH at the fourth, and the fifth it is connected to CH2NH2. Second, it is connected by O glycosidic linkage between the first and the fourth carbon. First carbon of your amino sugar, first ring, and fourth carbon of your sugar, deoxystreptomine. So what is 2-deoxystreptomine? It contains amino group at 1 and 3 position, no OH at the second position. So that is 2-deoxystreptomine. Fifth position OH and sixth position OH is connected by O glycosidic linkage to your first carbon of your another third ring amino sugar. Okay, remember at the first. Now in the second position it contains an OH, third position NH2, fourth position OH, and the fifth position it is connected to an CH2OH. Okay, that is your normal amino sugar. That is your structure of kanamycin. It was obtained from Streptomyces canamacetus. Okay, the least toxic member in the market is kanamycin. It is used in the treatment of gastrointestinal infection such as dysentery, systemic bacillus infection caused by Klebsiella proteus, Enterobacter. For disinfecting of your ear before operation. Kanamycin can be used. So this is the structure. It contains two sugar moieties, that is kanamycin and deoxystreptomine. Now next is your amikacin. Next is your amikacin. It is a semi-synthetic derivative of your kanamycin. So kanamycin, there is a modification of your kanamycin A. So what is kanamycin? OH, the first amino acid is same, everything is same. Second is also deoxyglucose. Only difference is it is connected by, it forms an amidic linkage. So with what it is connected to, with by an amide. The first amino group forms an amide, that is CO. It is alpha. This is 4-amino 2-hydroxy butyric acid. So it is connected to 4. It forms an amide bond with 4-amino 2-hydroxy butyric acid. So that's the only difference between your kanamycin and amikacin. Rest all structure is same of your kanamycin. Only thing it is acylated at your amino group. That's the only difference between your amikacin and kanamycin A. It is used in Mycobacterium Yersinia tularensis, Pseudomonas aeruginosa. The suffix -ing comes because of its origin. Okay, as we explained in the. Now next is tobramycin. It is isolated from Streptomyces tenebroides. Okay, it is antibacterial against resistant Pseudomonas. So it is effective against Pseudomonas aeruginosa. Now what is the structure? What is the structure of tobramycin? It contains an amino group. If you see in the previous structure, the second position has an OH. Okay. Now let's go for tobramycin. In case of tobramycin, the second position is an amino sugar. Your second position is an amino sugar. Okay. There is no OH at the third position. Remember, and the fifth position, it contains CH2NH2. Please remember, it contains CH2NH. It's only CH2 written. It contains CH2NH2. Okay. There is no OH at that. So there are two differences in the structure. One is instead of your OH, you get NH2, and there is no OH at the third position. Okay. Second structure is same. It contains deoxystreptomine. It is connected to the same. The amino acid is the amino sugar, and the third ring is also the same. There is no change. Only difference in the tobramycin is NH2 instead of OH at the second position, and there is deoxy at the OH at the third position. That's the structure of tobramycin. Now comes the structure of gentamicin, isolated from Micromonospora purpurea. Okay. That's why the suffix mycin comes, because of its origin. They are active against urinary infection caused by gram-negative and Pseudomonas species. Now let us see the difference in structure. Similar to your tobramycin, it is NH2 at the second position. OH is not there at the third and the fourth. So there is no deoxy at the third and the fourth position. There is no OH. There is no OH at the third and the fourth position. Next is what? Your group which is attached to the same. It can be either substituted at the, there is a methyl substitution at the amino group as well as at the secondary carbon. Okay. So it was found that secondary amino group at the 6th NH2 is gentamicin has spatial hindrance. So it is resistant to your acetyl. So amino group at the 6th position enhances the activity. So in case of gentamicin C1, okay, gentamicin C1, it contains CH3 group at your 6th carbon also and at the amino group which is attached. So there is a steric hindrance at the amino group. Gentamicin C2, R1 is CH3 and R2 is NH2. Okay. While in C1A, it contains your R. So basically, you have to remember gentamicin C1. Okay, that is the most active species. Okay, it also contains a 2-deoxystreptomine. That is same. Now let's go for the third ring. Let us see the modification that till here all the rings contain amino group at the third position. But in this case of gentamicin, it has got an N-methyl group. It has got an N-methyl group. Okay, it is an secondary amino group. Now let's go for the fourth carbon. So it contains an axial OH, tertiary alcohol, and a methyl group. And there is no CH2OH in the fifth position. Okay, so that's the difference in your structure. That's the difference in your structure, okay, of your gentamicin. Now let's go for the structure of neomycin. Let's go for the structure of your neomycin. It was isolated from Streptomyces pradea along with antifungal substance Facidin. Okay, this is also used on, it is used for your topical use basically. Now let's see the structure. It has got amino group at the second and the sixth position. So amino groups are present at the second. Similarly, it contains a deoxystreptomine. Now, one of the most important thing in this structure is that they contain an extra ribose sugar. So in the SAR, we said that some of the aminoglycosides contain extra sugar, extra D-pentose sugar, so ribose sugar. So this neomycin contains an extra sugar molecule, ribose sugar, 5-membered pentose sugar. Okay, 5-membered, which is connected at the first O-glycosidic linkage to your deoxystreptomine, and at the third position it forms a O-glycosidic linkage with your third ring. So similarly over here it contains your amino group at the second, third position OH, fourth position, and the fifth position contains your CH2NH2. Contains your CH2NH2. So this is the structure of your neomycin. What does it contain extra? Here it contains an, instead of your OH, it contains an NH2 in your fifth position. That's the major difference between the structure. That is known as neosamine. Okay, this is the structure of neomycin. Next is your netilamycin. So when we were studying the SAR, we said that netilamycin is formed when there is no OH at the third position, and at the fourth and fifth position there is a double bond. There is a double bond. Amino group at the second position. That's the basic structure, double bond. Netilamycin contains a double bond between fourth and fifth, no OH. So it is resistant against the deactivating enzyme because there is no OH or amino group. Still the activity is retained. Amino group is present at the second position, which is necessary for the activity. So next is ethylation. So your netilamycin in your streptomycin, it is a deoxystreptomine group, okay, which contains amino group. Only difference over here is your amino group in your deoxystreptomine contains a methyl group, okay, contains an ethyl group, okay. So if it does not contain ethyl group, it is called as cisomine, and if it contains an ethyl group at your amino group at the first, it is known as netilamycin. The rest all structure is the same. Similarly, when you go to the third ring, it is a secondary amine. It contains an methyl. It is similar to, see this structure. It is similar to your gentamicin structure. It is similar to your gentamicin structure. And methyl OH, CH3, OH is axial, and there is nothing at the fifth carbon, okay, of your third ring. Next is your paramycin. Next is your paramycin. It was isolated from Streptomyces rhamnosus. It was isolated from Streptomyces rhamnosus. It was used for the treatment of GI infection caused by Shigella, Salmonella, E. coli, and Amoeba. Now let's go for the structure. So this structure, it resembles to what? Your neomycin. It resembles to your neomycin. Okay. It contains, the first ring contains a NH2OH and CH2OH. That is a glucosamine. This is connected to a streptamine. Similarly, it is connected, it has an extra amino group. It contains an extra ribose sugar, which is connecting to your aminocyclitol to your third ring. So there is an extra ribose sugar. It contains a neomycin B or C. So that is your NH2 OH OH, and your fifth position. Okay, your fifth position contains your NH2. There can be a reverse, either it will be an axial or an equatorial. So only thing you have to see, when it is in axial position, it is paramycin 1 CH2 NH2, and when it is in equatorial position, it becomes paramycin 2. Only thing is the position, axial and your equatorial position, that changes the structure. That's the structure of your paramycin. So it contains an extra ribose sugar. The third position contains your amino, and rest all things are same as that of your neomycin. So this is your aminoglycosides. These are the amino. So the next amino acids that we have to study in detail is streptomycin. The next class of amino acids that we have to study is streptomycin. So streptomycin was first isolated from Streptomyces griseus. It was introduced against tuberculosis in 1943. Along with kanamycin, amikacin are effective against tuberculosis, but not as much as streptomycin. So streptomycin was more effective in case of tuberculosis. So this work of streptomycin gave Waxman the Nobel Prize in 1852. Now let's go for the further details of the structure of streptomycin. So basically, what does it contain? It contains a streptidine moiety. The first ring over here is streptidine, followed by a ribose sugar, that is streptose, followed by an N-methyl glucosamine. And this combination of your streptose sugar and N-glucosamine was constituted your streptobiosamine. They were linked to each other by your glycosidic linkage. The first is your streptidine. So in this case, your first ring is not your amino sugar. It is an aminocyclitol, followed by a ribose sugar and a glucosamine. It is a glucosamine. So this is the change in your streptomycin. Now let's go for the detailed structure. So first one is your glucosamine. At 1 and 3rd position, there is your guanidine linkage. It is 1,3-diguanidine linkage is present. That is NHC double bond NH, NH2 at the 1st and the 3rd position, and OH at rest of the position. Okay, 2, 4, 5, and the 6th position forms a glycosidic linkage with the ribose sugar, that is streptose. Okay, which contains your methyl at the 1st, an aldehyde and an OH. So CHO, that is an aldehyde and OH at the 2nd carbon, and at the 3rd carbon it forms a O-glycosidic linkage with your N-methyl glucosamine. What is this N-methyl glucosamine? It contains N-methyl group at your 2nd position. It contains an N-methyl group at a 2nd position. This is an example of amino sugar. So it contains a guanidine, streptidine moiety, followed by a ribose sugar and a N-methyl glucosamine. The ribose sugar is known as streptose. The ribose sugar is known as your streptose. Okay. So now let's go for the SAR. What do you mean by SAR, structural activity reviews? So any modification, whether it can be tolerated and what should be the important things for your activities is understood by SAR. Now modification on the streptose has extensively studied. So your streptose means your ribose sugar. Okay, the pentose sugar which is that is studied. So reduction. So it has an aldehyde group at the second position. It has an aldehyde group at the second position. If it is get converted into a reduced to an alcohol to form dihydrostreptomycin. So to form a dihydrostreptomycin, the activity has similar to your streptomycin but has severe deafness. DEAF. Okay, deafness. It causes severe deafness. Second thing, once reduction, side effect was more. Now when you oxidize, your aldehyde gets oxidized to what? An oxide. When you convert your aldehyde to an oxime, you like phenylhydrazone, results in inactive compound. So if you try to modify your aldehyde to any oxime, okay, like and hydrazone. Hydrazone is nothing but phenylhydrazone or PH NH NH2. When phenyl ring attached to a hydrazine is, and when it reacts with an aldehyde, it leads to a formation of your hydrazone. So when the aldehyde reacts with a phenylhydrazine, you get a phenylhydrazone. Such compounds also results in inactive compounds. Now oxidation of CS3, okay, to CS. At the second position, you know there is a methyl group of your ribose sugar. So if it is converted to your CH2OH, gives an active molecule but has no advantage over your streptomycin. So your methyl group at your second position of your ribose sugar, if it is converted into an alcohol, it is active but it does not have any much further advantage to that of your streptomycin. Now N-methyl glucosamine is essential for your activity. Your N-methyl glucosamine, that is your third ring, is necessary for the moiety. If you modify your amino-methyl group in a glucosamine by demethylation or replace it by alkyl group, higher alkyl group, the activity is lost. The N-methyl glucosamine, the nitrogen atom should always be secondary. Should be always be secondary and it should be methylated, no higher alkyl groups. The guanidino group in the streptidine ring are essential for activity. Change in anything, if you try to convert it into any other group, if you try to convert it to any other group, the compound loses activity. That is, the streptomycin loses its activity. Okay. So any change, okay, any change, if you convert your if you convert your guanidine, if you change that structure, okay, the activity is completely lost. Your activity is completely lost. So this is the SAR, very important of your. Now mechanism of incompatibility of your aminoglycosides with beta. Why should we never prescribe beta-lactams along with aminoglycan? There is an chemical incompatibility. So if you study it, your nucleophilic attack on your carbonyl carbon leads to your destruction of your beta-lactam ring. So and if you know, this aminoglycosides contain free amino group. In your cycloaminitol, it contains free amino group. So this will act as a nucleophile. It will attack on the carbonyl carbon of your beta-lactam, breaking of ring, opening of the ring, thus leading to the inactivation of your beta-lactam antibiotic. So never ever prescribe an aminoglycosides with a beta-lactam antibiotic. It destroys the activity because it destroys the beta-lactam due to nucleophilic addition across the carbonyl group by your amino acid in your amino sugar. So this is why you should never ever use, okay, your aminoglycosides with your beta-lactam. So this is a contraindication. Never prescribe aminoglycosides with an beta-lactam. Now some of the side effects. The most side effect is ototoxin. Netilmicin is reputed to be the mildest of the both. Okay, so they have effect on the ears. They cause nerve deafness, okay, of your auditory nerve. So they are also nephrotoxic. They are toxic to your kidneys also. They also have other neurotoxic effects like optic neuritis, peripheral neuritis, neuromuscular block. So you have to prescribe, because this will lead to your lot of malfunctioning in our body. Others are amyloedema, skin rashes, blood dyscariasis, eosinophilia, fever, stomatitis, anaphylaxis. So this can lead to anaphylactic shocks also. So, okay, blushing and all on your skin. So these are the side effects of your aminoglycosides. So they should be used in caution in case of pregnancy, myasthenia gravis, renal impairment, Parkinson's disease, and eighth cranial disease, because it affects your neuromuscular junction. So that's why we have to prescribe this aminoglycosides with caution. Okay, we have to prescribe this amino acids with caution.